Good afternoon. First of all, I would like to thank the organizers for the invitation to give a talk at the Congress. I'm going to talk about the subject which may sound fairly fundamental, but it may have serious translational implications for designing vaccines. The subject of the study in my group are follicular regulatory T cells and how they control B-cell responses, including B-cell and antibody responses to vaccination. As you know, many vaccines are aimed at generating long-term high-affinity antibody responses against pathogens or specific antigens on the pathogens. And for this, antigen-specific B-cells usually need to be recruited into T-dependent B-cell response and enter into the so-called germinal center B-cell reaction, where antigen-specific B-cells undergo rapid proliferation, somatic hypermutation of their B-cell receptors, and affinity maturation. So in this process, B-cells which have B-cell receptors or immunoglobulins with a higher affinity for antigens can generate memory B-cells and plasma cells, including long-lived plasma cells that produce higher affinity antibodies. This process of selection of B-cells that acquire higher affinity to antigen requires help from the follicular helper T cells that are specific to foreign antigens. However, this is a very complex process and multiple things can go wrong, including recruitment or generation of autoreactive B-cells in this process. For example, B-cells that have cross-reactivity to foreign and self-antigens can be recruited into dependent response and generate short-lived plasma cells. But in addition, some cross-reactive autoreactive B-cells can be generated within germinal centers due to the random process of somatic hypermutation of the B-cell receptors. And these processes have to be tightly controlled. Indeed, there is a subset of cells which is involved in this process. In general, regulatory T cells are controlling autoreactive B cells, but also there is this subset which is called follicular regulatory T cells. Follicular regulatory T cells are FOXP3 expressing Tregs that also acquire expression of the transcription factor BCL6 and upregulate expression of the chemokine receptor CXCR5, which enables them to go into B-cell follicles and under certain conditions enter germinal center responses. In mice, in the early stages of the immune response, majority of follicular regulated T cells derive from natural thymic-derived Tregs, many of which are specific to autoantigens. However, until recently, it was unclear what kind of antigens can trigger follicular regulatory T cells in a T cell receptor dependent fashion. In the recent study, we have hypothesized that nuclear proteins, which are often targeted by autoantibodies in autoimmune diseases, may contain autoantigens that could trigger TFR. We have then shown that germinal center B cells that acquire nuclear proteins can trigger very rapid accumulation of follicular regulatory T cells in mice, which in turn can suppress participation of these B cells in the germinal center reaction. Follicular regulatory T cells have attracted a lot of attention in the recent decade. It has been shown in multiple studies that they can moderately suppress foreign antigen-specific response at certain stage. They are required for negative control of autoreactive B-cell responses, so that deficit in follicular regulatory T cells leads to generation of autoantibodies and autoimmunity in mice. But from the other side, follicular regulatory T cells somehow assist in the process of affinity maturation of germinal center B cells. germinal centers is a very complex dynamic system, right? Which is thought to be divided into two zones, so-called light zone and dark zone. Within light zone, germinal center B cells compete for foreign antigens on follicular dendritic cells and help from follicular helper T cells. Those B cells that are selected by these two signals then get into the dark zone, where they start expressing transcription factor FOXO1, which enables them to undergo proper proliferation and somatic hypermutation for the next round of return into the light zone and for the next round of selection. In this process, some of the selected germinal center B cells also form memory B cells and long-lived plasma cells. Follicular regulatory T cells have been shown to have a suppressive effect on both B cells and follicular helper T cells. However, interestingly, some studies demonstrated that IL-10 produced by follicular regulatory T cells is necessary for expression of FOXO1 transcription factor in germinal center centroblasts, therefore supporting the cycle of B cells within germinal centers. However, until recently, this dual role of TFR cells in the control of B cell response to multiple vaccinations or diseases in mice was unclear. We were able to look into the mechanisms of this dual regulation of B cell responses by follicular regulatory T cells due to our earlier discovery that a certain chemokine, CCL3, which is produced by activated B cells and a subset of germinal center B cells, promotes their interactions with follicular regulatory T cells in a chemotaxis-dependent fashion. In that initial study, we have shown by imaging wild-type B cells shown in blue together with CCL3 deficient B cells shown in green within germinal centers of mice, and by analyzing their interactions with follicular regulatory T cells shown here in red. We performed very careful analysis in 3D of the interaction of follicular regulatory T cells with CCL3 proficient and deficient germinal center B cells, quantified the number of contacts and normalized them to the number of B cells of each kind present in the germinal centers. By this analysis, we have been able to show that CCL3 deficient B cells have reduced capacity to form contacts with TFRs, and this is in live mice. So CCL3 really promoted the contacts between bulk germinal center B cells and follicular regulatory T cells. This effect we have not observed for follicular helper T cells. So the next question was, if CCL3 promotes the interactions between TFRs and germinal center B cells, what is the functional significance of CCL3 expression by germinal center? To address this, we have utilized both bone marrow chimeras model and adoptive transfer model, which I will describe later. In the bone marrow chimeras model, we have co-transferred bone marrow from CCL3 deficient uli-terminal control mice together with CCL3 proficient CD45.1 bone marrow into irradiated recipient mice. After reconstitution of the bone marrow, these mice were immunized with a very well explored antigen in mice, NP HAP10 KLH. And after the immunization, the lymph nodes were collected for flow cytometry analysis at various times after vaccination. We have utilized the flow cytometry gating strategy to look at both the germinal center B cells, which are fast high, CD38 low cells in mice. We could distinguish CD45.2 cells or CD45.1 cells within germinal centers, and also look at the germinal center B cells which were specific to NP or could bind NP antigen with high affinity and in a way represent the process of affinity maturation of the cells. We also looked at the formation of the memory B cells which were specific to NP. This led to two effects which we could observe. First, formation of memory cells by CCL3 deficient B cells was reduced, and accumulation of germinal center B cells with higher affinity to antigen was also reduced. We confirmed defects in the affinity maturation by the analysis of the B cell clones that should have acquired high mutation that should have higher affinity to NP antigen. To summarize, we have shown that expression of CCL3 by germinal center B cells was required for their prolonged participation in the germinal center response, formation of memory cells over time, and affinity maturation. We then asked whether this process was dependent to TFRs, as CCL3 promotes B cell interaction with follicular regulatory T cells. And in this case, we have utilized an adoptive transfer model where mice were co-transferred with transgenic B cells specific to DACA glycozyme model, which either express CCL3 or were CCL3 deficient. Once again, mice were immunized, and we could track participation of both types of cells, CCL3 proficient and deficient, in the immune response over time. Consistently with the bone marrow chimeras model, we saw that CCL3 deficient heightened B cells dropped out of the germinal centers much more quickly. We then could transfer the cells into mice which were deficient for follicular regulatory T cells. This is BCL6 FOXP3 model. And you can see the drop in the follicular regulatory T cell numbers compared to the control mice right here. While in mice that had normal amount of TFRs, CCL3 proficient cells stayed longer in germinal centers than CCL3 deficient. This difference was abolished when follicular regulatory T cells were not around. Therefore, not only CCL3 but also follicular regulatory T cells support participation or prolonged participation of B cells in the germinal center response. In the previous study, it was suggested that IL-10 produced by TFRs is important for their participation in the dark zone germinal center program. So we then have asked whether this effect was germinal center B cell intrinsic and whether it depended on the expression of IL-10 receptor on germinal center B cells. To address this, we have generated bone marrow chimeras now with B cells, half of which lacked IL-10 receptor and half of them had the wild-type levels of IL-10 receptor. Mice were once again immunized, and we looked at the germinal center response, memory cells, and affinity maturation over time. As expected, over time, IL-10 receptor deficient germinal center B cells formed fewer NP-specific germinal center B cells. We could see that while at 10 days after immunization, participation of IL-10 receptor proficient and deficient germinal center B cells was comparable. At 18 days after immunization, IL-10 receptor deficient germinal center B cells were almost gone. This resulted in the defect in the affinity maturation of the cells and deficit in the formation of memory cells. However, in the control cells and the control chimeras, this effect did not occur. So, to summarize this, our study has suggested that CCL3 promotes interactions between germinal center B cells that produce CCL3 and follicular regulatory T cells. These interactions are required for sustained participation of B cells in the germinal center response and requires expression of IL-10 receptor on germinal center B cells. So this shows that these direct interactions are required for sustained participation of germinal center B cells in germinal centers and proper affinity maturation. However, previously we have shown that when germinal center B cells acquire nuclear antigens, nuclear proteins, TFRs suppress them. So then we have asked whether direct interactions in a CCL3 dependent fashion are important for this, or the mechanism is distinct. And once again, we have generated a mixed chimeras model where half of B cells expressed CCL3 and half of B cells were CCL3 deficient. These B cells were then immunized with streptavidin to promote accumulation of streptavidin specific germinal center B cells, and boosted with streptavidin or streptavidin linked to the selected nuclear proteins. In the previous study, we showed that this boost results into the increase in the follicular regulatory T cell numbers. Once again, we observed a very significant effect within 3 days after the boost. If we look at the control cells that were CCL3 proficient, these accumulating TFRs suppressed germinal center B cells that acquired nuclear proteins. However, when B cells lacked expression of CCL3, follicular regulatory T cells were unable to suppress participation of these germinal center B cells in the immune response. Therefore, CCL3 and direct interaction between germinal center B cells and TFRs are also required for direct suppression of those germinal center B cells that acquire nuclear proteins and present this nuclear protein peptides for follicular regulatory cells, presumably in a MHC2 dependent fashion. To summarize overall this observation, we suggest the following model of the germinal center B cells dual control by follicular regulatory cells. We suggest the germinal center B cells, which undergo selection in the light zone, upregulate expression of CCL3 and 4, and we show this in our study. There is no time to talk about this, which then attract attention of follicular regulatory T cells in a chemotactic fashion. The interactions, which are not necessarily cognate between germinal center B cells and TFRs, are nevertheless sufficient to supply the B cells that undergo selection with IL-10 produced by TFRs through IL-10 receptor, which then promotes FOXO1 expression in germinal center B cells and their prolonged participation in the dark zone, proliferation, and participation in the immune response. However, if germinal center B cells become cross-reactive to self-antigens, for example, nucleosomes or other outer nuclear antigens, this can lead to the cognate recognition of the TFRs that sample germinal center B cells through T cell receptor peptide interactions, MHC. And these TFRs then will suppress germinal center B cells that become autoreactive. Therefore, TFRs act as a dual control of the B cells that are undergoing selection. With that, I want to thank the people who have done this work, talented postdoc Funke and former graduate student Zachary Bennett, our collaborators, the labs who have supplied mice, and colleagues who have advised us on these experiments, and of course our funding. Thank you for your attention.